Hello, everyone. Welcome to the Thomas Expert Series. Thank you for joining today. We're going to get started in just a minute, just to get everyone a little warmed up. Want to see where everyone is tuning in from. And while you're doing that, please make sure your microphones are muted. See where we've got people coming from today. Got Colorado, Philly, Jersey, Tennessee, some more, Phoenix, West Virginia. Got a lot of East Coast in here today. South Carolina, Florida. Oh, Vancouver. Catherine, where are you at?
I'm just outside of Baltimore, Maryland.
Nice. So I'll go ahead and turn it over to Catherine here today to talk about innovative technologies for creating 3D cellular models.
Excellent. Well, thank you for joining me, everyone. Happy Friday to you. Hopefully this is on the downhill for your Friday. So I am going to share my screen here. And we will get started and honor your time. So we're going to talk about 3D cell culture today. Then the presentation mode. Okay, and then Heather, can you confirm you're just seeing the one slide?
Yep, looks great.
All right, perfect. So let's start talking about 3D cell culture. So this is something that Corning has been pretty focused on for the past decade. So if you're new to it, I'll give you a little background and we'll talk about some of the things that we like to offer to customers to help them start down this path. So the plan is we will talk about 3D cell culture. So why do we even have this as a thing? So we'll talk about the need for a cell culture model that is more realistic. We'll talk about taking those cells, those cellular models, into the third dimension, and then we'll talk about scaling up those 3D workflows. So really, this all starts with cells. Corning believes in the power of cells, and they are a really great experimental model. So, of course, organisms are composed of billions of cells, and they all perform really important functions in the body. So different cell types perform different functions. Our liver processes toxins. Immune cells are there to fight infections. And then lung cells help to get that oxygen back into the blood. So everybody has a pretty important job there. Now, the reason that we like to work with cells is because studying an entire organism is pretty difficult to do. Even something like, your typical lab mouse. There's a lot going on there. And so if we want to answer some questions, we got to simplify it. And so we go down to cells. Cells are great because we can grow these in dishes in the laboratory. We can control just about everything in the environment. And so that way we're able to answer questions because we're in charge of everything. So if I change one thing, what happens? That helps us make further discoveries in science. So the problem is that when we grow cells, we need them to represent whatever system we're trying to study, whether it's the lung, the liver, what have you. So for a long time, cells have been grown in this kind of 2D fashion that you see over here on the left. So that will give you the cells, and they will be touching some other cells, but you're not going to capture all of the interactions that they might have. So in your body, cells are in contact with all kinds of things. So not only cells of the same type, but cells of other types. So even in your lung, you think, okay, it's a lung cells. That's like 9 different types of cells, but they're all working together. They're talking to immune cells that are in the environment, and they're interacting with the extracellular matrix, which is like this protein network that's outside of cells, and it helps give them support. So there's a lot of interactions that we just don't quite capture if we're growing cells in 2D. So we want to try to grow them in 3D. So for a long time, cells have been grown in 2D. As I mentioned before, kind of just flat in a dish, and there is still utility there, but maybe for something a little simpler. So that's pretty easy to do. We can crank that out. Good throughput there, easy to do. Now, on the other end of the spectrum, if we need something that's a little bit closer to a human model, we could go to something like an animal model, like a mouse model. It is biologically complex, lots of different things interacting, but these are expensive and they take a really long time. And plus, they're not human. So we find some middle ground with these types of 3D cell cultures. So spheroids and organoids are things that are really prevalent in the field right now. And today we're actually going to focus on spheroids. So these are structures that are going to use human cells, and we are going to be able to kind of condense those human cells into a more 3D structure that's more similar to like an organ. So if we think about it as, all right, I'm in 2D, I need to have this more realistic system. How do I get them into 3D? Problem is that cells really like to stick to each other and they like to stick to the surface of the dish. But if we want to make this little solid aggregate of cells, we need them to only stick to each other. So what we have done there is modified the surface of that dish so that the cells can't grow on it anymore. And so the result there is that the cells stick to each other and they form those spheroids that we mentioned. Now that modified surface on our plates and dishes is called ultra low attachment. So our ULA surface is just like a gel that's already coated on the plate, and it prevents cells from binding to the plate. So if we look at our schematic on the left there, normally cells kind of spread out and they attach to that plastic. But then at the bottom, if we have our ultra low attachment surface on there, they can't stick. They kind of have to stick to each other. And you can see a real life example over on the right. So these are the same type of neural tumor cells. When we put them on a regular tissue culture plate, you can see they kind of stick and they flatten out. When we put those onto that ultra low attachment surface, you can see that they ball up and they form these little spheroids. So spheroids in particular, I've said that word a couple of times, but I wanted to give you like a quick piece of background on it. So spheroids are where, they're pretty aptly named, like it's a spherical little model, and it's something that we're going to kind of create. So we're going to take cells from a 2D plate and we're going to put them into an environment where they're going to form a 3D structure. So that's usually people working with like a cell line that they grow over and over again. And they really don't need any special help in terms of growth. So we're going to put them into a plate that'll help them aggregate, but then they usually don't need anything other than that. Just your typical media serum, don't need like a scaffold or any matrix. So we talked about ultra low attachment surface. That is the way that we're going to get cells to not stick to the plate, but rather to each other. Now, the problem there is that if we just put some cells into that ultra low attachment plate, it can form different sizes of spheroids. And if we're trying to study how a drug works on a particular cancer line, we need everything to kind of be the same size because a drug might work well on a really small spheroid, not so well on a big one. So really consistency is going to be the key here. So the solution is combining our ultra low attachment surface with some special geometry to help encourage really consistent spheroid formation. So our comparison down on the bottom here, if we have just a typical 96-well microplate format with our ultra low attachment surface, we got lots of spheroids in the well. So, okay, we've got a spheroid, that's good, but we want consistency. So here is where we add in a little bit of special geometry to help get one even spheroid in every well. And that is our spheroid microplate. So this has a clear bottom. So the shape of the bottom helps those cells coalesce together. You get our one nice little even aggregate in each well. Clear bottom, so you can visualize it. And you can do a lot of different types of assays in here. So people are going to grow these cells, but then they want to see how they respond to different things. And so they're going to do that by looking at different signals that the cells put out. So they can do fluorescent and luminescent assays all on the same plate. Grow it, assay it, plug and play. Now, what if we need more? Often the next question in science is, well, that's great, but I need to scale it up. How are we going to do that? So sometimes a researcher might just need more data points, or they would need to run more experiments, but I don't want to go through dozens and dozens of plates as much as we would like to sell them all those plates. We do have a solution for that. So what if we just modify the plate? So instead of one spheroid in every well, what if we could have hundreds or thousands of spheroids in every well? We're going to do that with microcavity technology. So this is our L-plasia product line. So it's a weird name, but a really great technology. So it has that same ultra-low attachment surface that we talked about before, but in this microcavity format. So you can almost think of it as kind of like a little honeycomb structure. So little microcavities where you get one spheroid in each of those microcavities. But in each well of these multi-well plates, you can get dozens, hundreds, thousands of spheroids in each one of those wells. So for something like this, people are going to be doing things like drug discovery, maybe doing some genomic screening, or maybe some cell therapy, something where they're going to need lots of spheroids in one vessel. And as I just said, sometimes the question in science is, well, that's great, but I need more. And that's kind of what happened here. So researchers came to us saying, like, I need to create thousands of spheroids at one time. I can't get by using these multiple plates. What are we going to do? We have a 12K format of L-plasia, 2 actually. So 12K is an appropriate name because we can grow 12,000 spheroids in one vessel. So we have this flask format, like a T75 culture flask format, but with that microcavity surface on the bottom that lets us grow those 12,000 spheroids in one environment. Our newest format here is the open well plate. So that's a microplate format. So we talked about like the 96 well plate before. This is basically a 12,000 well plate, but in that same format that can go into robots and liquid handlers. And so this one in particular, you can start to plug into some automation workflows. So to sum up what we've talked about today, we have several great options for researchers to grow their spheroids in a scaffold-free and assay-ready format. So the foundation of all of these products is our ultra-low attachment surface for the spheroid plate and for all of those L-plasia formats. So spheroid microplates are going to allow researchers to grow one spheroid in each well of the plate, whereas our L-plasia microplates allow researchers to grow multiple spheroids in each well of the plate, and then our 12K plate and flask formats allow researchers to grow 12,000 spheroids in one vessel. So one final point that I want to make before we get to our Q&A for today, it's a great time of year to sell fetal bovine serum. So as academics come to the end of their fiscal year, sometimes they have a little cash that they've got to burn, and we have really great supply of fetal bovine serum right now. So if people are doing cell culture, that means they need flasks, they need media, and they need serum, especially if those cells are growing attached to the flask. Just about everybody is going to use serum for that purpose. So we have a huge variety of the FBS, lots of different types. People can test it if they prefer, but our supply is great and it's a great time of year to sell it. We also have a really good promotion on it. So if people purchase 10 bottles, they can actually get one for free. A promotion like that is a little unheard of, but it's a great time to offer that to your customers. All right, so that's all the content that I have for today, but I would love to hear your questions. So let me go ahead and stop sharing this. We can always go back if we need. But let me know what you would like to know, or if you have customers that are working on this type of stuff, what can we do to help you?
And while everyone is typing out their questions, I want to hand it over to Beth, who is going to take you into the environment for just a moment.
Okay, let me share my screen. We'll start out in the expert series. And as you can see, here's the Corning kiosk. Welcome greeting right here. So check that out, a little bit about Corning. And of course, the resources that they filled for the month. This will be up the entire month of June, literature PDFs, web links, and videos. You can download, print, or add to your virtual briefcase. And now I'm going to take you into their lab. You can go directly this way. And the Corning Lab has a lot of the products that we were just talking about, especially the serum right here. So each one of these pluses, please click on those for the resources as well as the signs. And they will be having office hours. So please talk to your Thomas rep, you customers that are on the call, and join them for the next Corning office hours. Do we have any questions?
Is somebody going to read the chat?
We can do that.
Yeah.
I'll volunteer.
Okay.
All right. Does Corning still produce the EPIC technology utilized for drug discovery?
We do not, unfortunately. So that was discontinued a couple years ago. It was a technology that worked really well, but it was kind of like a niche product. And so it worked really well for a handful of customers, but just it never got the reach that we wanted it to. So we discontinued that a couple years ago, unfortunately.
And how do you control the size of your spheroids?
Yeah, that is a good question. So it really comes down to just how many cells you put into each well. And the size is something that we really ask customers to think about, because you can imagine if I'm studying tumors, well, tumors have kind of like an outside and an inside. And at the inside, they don't get as many nutrients as the cells on the outside. You can start getting some cell death in there. So if I want a tumor model, then I probably want a spheroid that's a little bit bigger. And so I'm going to put more cells into every single well. But if I want this to be kind of like a tissue model, like I want this to be a nice little mini liver for me, I don't want it to be vastly different from the outside to inside. And so I'm going to make that a smaller spheroid. So that is something that we can pretty nicely control.
Okay. And there's quite a few here. How do you make sure the spheroids don't get displaced when you're doing something like a media change?
Yeah, so for the Alplasia products in particular, for both the 12K flask and the plate, we did some engineering to kind of prevent the turbulence when you're doing a media change. So there's some design features in there that make sure everybody stays in their little nest. For the other Alplasia products, it's a matter of just keeping the media volume a little bit higher and then doing like a half media change. So instead of going way down to, a dry plate, just changing out about half of that volume.
Okay. Next question. Do you make a 384 PCR? It keeps moving as I'm talking. PCR plate. Sorry.
Genomics is not necessarily my area of expertise, but I want to say that we do. We do have a pretty comprehensive portfolio. And if I'm remembering correctly, I think we even have a new hard shell version of the 384 well PCR plate. Sometimes during PCR, the plates can kind of warp a little bit, and then you just get some inconsistent results. So with that hard shell plate, you get much better consistency performance throughout the cycle, because of course we're heating and we're cooling. So plastics, that can make it a little uneven, but that hard shell plate has good performance. And I'm 90% sure we have a 384 in that.
And then just to tack on to that, are there smaller formats of the spheroid plate?
Yes, there are. So I showed the 96 well there because I think it's a good illustration of just kind of what it looks like. But there is a 384, and then there's also a 1536. So if people are doing like super high throughput stuff, automation, there are some plates that can plug into those workflows for sure.
And another question, is agitation/circular motion required to have the spheroids form correctly?
No, that's the nice thing about the spheroid plates. It's really the combination of the ultra low attachment surface and the special geometry of the plate that just kind of force the cells to come together. It's almost like a little teardrop geometry at the bottom of the plate. A little bit different than a typical like round well bottom. This kind of like concentrates everybody down in the bottom. So you don't have to agitate it, which is great.
Okay, another question. What are some applications or processes that people are, sorry, it just jumped, doing with the microcavity plates?
Yeah, so for the alloplasia plates, people are doing some pretty cool stuff there. So sometimes it's drug discovery, where we have a cell line, we have some small compounds. We want to see if maybe it's a tumor cell line, and we want to see, does this kill the tumor? Does it have any effect? Or we have this drug that we think is going to work. Let's put it on other cell types that we find in the body and make sure that it doesn't kill that, like especially with liver cells. There's plenty of drugs out there that might have a great effect on a tumor, but it's also very toxic to the liver. So that's a lot of what people are doing with drug discovery. People are also using them to create extracellular vesicles. So cells are always producing something, and they butt off a lot of these little membrane things, extracellular vesicles, which can be really helpful for diagnostics. So there's a lot of work going on for just what are cells telling us on a regular basis with these little kind of messengers that they're sending out. So drug discovery, extracellular vesicle production, even like tissue engineering. We have people working with them to create some skin therapies for people that have a congenital condition.
Do we have any other questions? Let's see, did something just pop in there? Do you provide samples of these pieces for customer evaluation?
Yes, Oscar, we do. We do have samples of these available. And so we'd be happy to talk to you about getting that into the hands of your customer, and then also setting them up for success.
Plates.
So getting them the sample, and then talking to them about like, what are you working on? How can we make sure that this is going to be successful for you? But definitely an option to have a sample.
Okay. Any other questions?
It's late afternoon on Friday, so if everyone's brain is a little fried, so is mine.
All right, if there are no more questions, I'll go ahead and close this out real quick. I just want to say thank you, and this talk will be on demand within 24 hours, so please let everyone know how much you enjoyed the talk today, and if you have any other questions, feel free to reach out to your Thomas rep.
Thank you, everyone.
Thanks.
Thank
Have
you.
a great weekend.
Have a good weekend.
Take care.
Take care.